specifically, what I'm going to be talking about is a process that's come to be called the rhizophagy cycle, and this is a process where plants internalize microbes, fungi and bacteria into their tissues, endophytes, but further to make them endophytes, but further they actually will incorporate them into the root cells, internalize them, and degrade some of them to extract nutrients from those microbes. And okay, so in this process, I think you'll see that, and I will try to convince you that plants are actually farming these microbes in the soil. So just like we farm or cultivate crops, plants, the plants themselves are farming or are cultivating these microbes. And the point of this whole process, the rhizophagy cycle, is so that plants can actually get nutrients from these microbes. So endophytes, I think most of you are familiar with what an endophyte is. Okay, it's a microbe that goes inside a plant, and they typically go into plants, or plants take them into their tissues, and they cause no disease. You know, so we have pathogenic microbes that are disease agents, and then we have endophytes that are non-disease agents. And you've heard probably about the fungal endophytes of grasses like fescue and ryegrass. And this image actually shows hypha of one of these epichloi or grass endophytes, stained blue with aniline blue, inside tissues of the grass plant. Okay. But all plants have endophytes, not just grasses. And actually they can be bacteria, and they can be fungi, and they tend to be not just one species in plants, but communities, multiple species that go into plants. Okay, so plants have all of these different microbes that go into their tissues and into their cells, and we don't normally think of that. We think of, well, a plant cell is going to be sterile except for the plant cell, unless there's a pathogen there, and that's just not true. Healthy roots and healthy plant cells in general, even in the shoots, will have endophytic microbes either in the cells or between the cells. Okay, so here's a plant. And I have one of my graduate students is April Michi, and she's working on hemp endophytes. And you think, well, hemp has been cultivated for a long time, and certainly, it's pretty clean. People grow it indoors and in under clean conditions. But in fact, if you look at the hemp seeds and germinate the seeds, those seeds are really filled with microbes. And you can see to the right here, that's a root hair of a hemp seedling. And you see all those little brown circles inside there. Those are all microbes that are inside that root hair in this case, but other root cells and other parts of the hemp plant also contain these microbes, multiple species. These happen to be bacteria, but they're in there. Any plant you pick will have these microbes inside their root cells and inside their other cells. Okay, this is also hemp again, and you can see here all this brown. This was stained with a stain to show the actually hydrogen peroxide around the bacteria. You see all the brown there, that's all hydrogen peroxide. This is in the root hair, and you can see the little blue lines in there as part of this. Let me see if I can get an arrow there. You can see that around okay, those are the bacteria inside that root hair. So these bacteria are coming off of the seed or from within the seed, and so they don't have to come out of the soil. Okay, but in fact, the plant takes the certain ones that are seed vectored, puts in the seedling, but it also will fish microbes out of the soil. So it's important to have a healthy soil because those microbes are important for plant growth. Here's another plant. This is from the desert island of Bonaire in the Dutch Antilles, and these giant cacti called Kadushi cacti, and they have little fruits, little purple fruits that go up way up high. But you take those, pull the seeds out of those fruits, germinate those like this. You see here the seedling. This is an agarose germinated in pretty much azenic conditions, or without soil, without all the other microbes. So that agarose is clean. It germinates, and you look at the root hairs. We stain them, and you can see the bacteria are already in that seedling. So these plants coming out of the seed will have these microbes with them. This shows some root hairs of that cactus, and you can see they're loaded. They're jam packed with these bacteria inside those root hairs. All plants have these kind of microbes. All plants that produce root hairs do this process called the rhizophagy cycle. Rhizophagy, rhizo for root, phagy for eating, and it's the idea that plants are actually eating these microbes. Some of the microbes vector on the surface of seeds, and so you have these tissues on seed surfaces where there's bacteria and yeast and so other fungi. And also some vector within the seed itself in the embryo. And mint is one of those. And these are some students who are doing this work on mint. Fernando, one of my graduate students, Gianna Pecorella, Charlie, one of my undergraduates. But they're looking at mints. And when you look at a mint seed, okay, here, you could even surface disinfect it to remove all the surface microbes, but you have inside that seed, you have bacteria being vectored. And this is a typical ovule, kind of drawing an ovule. This tissue, the central tissue there is the embryo sac, essentially. There you can see the very early, it has a nucleus there. You see they're labeled, and that's a typical embryo sac. And then you have this cellular tissue that goes around that. This is all part of inside the seed. This is called the nucellus. That nucellus is actually full of bacteria. These other tissues on the outside, integuments, they become part of the seed coat out there and form a, part of that seed coat itself, the external tissues. But the bacteria in that nucellus, and we'll see images of that. And here you can see the outer tissues here, the rough tissues to the left. And then you see right into the right, you see cellular nucellus layers. It's that layer that has the bacteria in it, and that is right near. Okay, here you can see one of these nucellus cells, and you can see this bacteria. You can see the arrows indicating these bacteria that are in that tissue. And it's this tissue that surrounds the embryo sac. So what happens when the seed germinates, the embryo will come out of that embryo sac, and then these bacteria, even before it gets out of the seed, will colonize that seedling that's coming out. That's just close up those bacteria. Another close up. Okay, and I mentioned that these bacteria essentially come from soil. This is the first source of these bacteria that plants get. So the plants will fish these bacteria out of the soil by attracting them to the plant, and then they will put them on the seed or in the seed. And so that's essentially how. So seeds come with these microbes in them, but the first source is always the soil. So that's the importance of having a healthy soil with lots of microbes there, good diversity of microbes in the soil. And okay, so what do these endophytes do? They will actually improve the stress tolerance of plants. They will make plants more resistant to heat and heavy metals and salt, for example. Anything that's oxidative stress of a plant, they will actually improve the resistance to. And the reason they improve resistance to oxidative stresses is because they affect the oxidative, they're part of the oxidative interactions. The plant actually uses reactive oxygen to interact with these microbes, and I'll explain it more detail. But that makes the plants resistant to oxidative stress. So they make plants hardier. They will also, these bacteria and sometimes fungi will also suppress pathogenic fungi. Some of these microbes will go out into the soil and they'll colonize these fungi and they'll make them avirulent. So they don't kill the fungi, but they affect the behavior of the fungi. They make them avirulent, they reduce their growth, they reduce their ability to cause disease. Don't kill it, but they make it so it doesn't cause disease. In some cases, those pathogenic fungi will actually turn into endophytes, helpful, beneficial endophytes when those bacteria are present. These microbes will also modulate root development and shoot development as well. In other words, the microbes are necessary for plants to develop properly, and I'll explain that as I discuss what they do for plants. They improve nutrient absorption. These microbes actually work directly for the plant in acquiring nutrients in the soil. They acquire those nutrients and they go back to the plant, and then the plant takes those nutrients from those microbes. And so the plant is using those microbes to get nutrients out of the soil. I mean, that doesn't mean that that's the only place plants get nutrients from. Some nutrients are dissolved in the soil water, and so plants can absorb those. But other nutrients are actually carried by these microbes. So I'll explain that. Okay, and these microbes in plants, the endophytes in plants, will alter the chemical constituents of the plants. And I'll describe an experiment where we did with and without microbes. And with the microbes, you'll see that the plants we used were carrots, and carotene is increased in those plants. So the microbes directly will improve the chemical profile, or they'll alter the chemical profile of plants. Okay, so they have all these effects on plants. Back in 2010, some Australian investigators, this young lady here is one of those, Chani Pong Fu, in Queensland, Australia, were looking at some plants and doing experiments with plants, and they happened to find that some microbes, yeasts, and bacteria were going inside the plant roots. And what they did is they would label the bacteria with the GFP so that they could then see it. They knew which one they labeled, and so the bacteria would fluoresce when they looked at it in a microscope. And then they fed it to plants, and these plants took them into the cells. And this is an illustration from their work, and you can see over in B, image B here, you see a little root hair forming there, but those are the root cells, and those bacteria went into those root cells and into the root hairs. You can see also on D, all those hairs there, those are all the root hairs with the bacteria in it. And down at E, you can see all those little spherical things, that's a root hair with the yeasts in it. So they saw this process, and they actually wrote an article called Turning the Table, Plants Consume Microbes as a Source of Nutrients. And later on, they named this process rhizophagy. Rhizo, like I said, for root, phagy for eating. And that conveys the idea that these plants are eating soil microbes. And, we started looking at the process, and we actually have discovered that the process is cyclic. And that is, the plants don't just consume these microbes. Some of them get consumed, but some of them actually go back out into the soil. They're replicated inside the plant. They go back out into the soil. They acquire more nutrients, and they're attracted back to the plant. And I'll show this. You can see here the diagram of a root tip. Actually, you can see the meristem here to the right, and just behind the meristem actually is a zone where the root is secreting exudates, and those are sugars. They're organic acids, and they're amino acids. And those are nutrients that these microbes need. So the plant secrete those nutrients out, those exudates. The microbes are attracted to those exudates. The plant then internalizes some of those microbes into the root cells, just underneath where the exudates are being secreted. And then inside the plant hits the plant hits these microbes with superoxide. And superoxide is a very potent form of reactive oxygen. That superoxide will strip the cell walls off of those bacteria, make them form into naked protoplasts. And then it essentially extracts nutrients from the microbes. The microbes that survive will stimulate root hair development. Without the microbes, we get no root hair development. With the microbes, we get it. And and then as those root hairs elongate, the microbes are then ejected back out into the soil. And the plant will then put nutrients after the microbes, more exudates where the microbes were ejected, and then the microbes will reform their cell walls, and then they'll go back, or flagella if they produce flagella, and they'll go back, swim back out into the soil and acquire more nutrients, later to be attracted back to the plant at the root tip meristem. So this rhizophagy cycle happens at the root tip, at root tips, only root tips. So wherever you have root tips, you have rhizophagy cycle happening. And wherever you have root hairs, you have microbes being ejected into the back out into the soil. Okay, the picture here to the left, you can see a tip of a root hair. You see all the little fluorescing particles out there. Those are bacteria coming out of that root hair, being ejected from that root hair. And if you look to the right there, this probably is B. It's kind of covered, so I don't see what it actually is. But let's see if I can. Yeah, but anyways, those little they're little bacteria inside those root cells, and that's what that shows. Those bacteria entering the root cells, right there at that zone. Okay, this is a grass seed germinating. Grasses carry microbes on the surface, and when it germinates, those microbes come out and they colonize the root. And they also colonize the shoot, and they're usually several microbes, 3 or 4. There's a little community on those seeds. When the root comes out, the microbes will colonize the exudate zone just behind the meristem tip. And here you can see that root exudate zone, and that is actually where the microbes are being cultivated, in that zone. And here's an actual picture of a grass root tip, and you can see this cloud of blue dots out there. Those are the bacteria that are growing in that exudate zone, and they are right at the surface of the root. They're entering into those root cells. Now the root cells at that point have very thin walls. They're not hardened walls, so these microbes can get through that. We don't actually know the process, how they're getting through. It could be enzymatic, where they digest their way in and get in, or it could be some other physical process where they're pressed into the wall, or some people have hypothesized that these root cells are phagocytizing, or just consuming them, bringing in, endocytizing them, bringing them into the cells. We don't know how the mechanism is, but we do know that they go in because we can see them inside those cells. And once they go in, okay, they will actually lose their cell walls and become protoplasts. And this kind of illustrates that. These are regular bacteria, and you can see the rods. Okay, those have cell walls. And what happens when they go in, the plant cell detects them and shoots superoxide on them, which oxidizes away the cell wall. And then they form these protoplasts. And you can see to the right, these protoplast phases. And this is just a phase of a bacterium without the wall there. And when you remove the wall, these protoplasts can be replicated very fast. And that actually is what happens once the plant has these bacteria inside those root cells. They replicate them very fast. Besides removing the cell wall, then they're replicatable, and their nutrients are extractable easily from them. The plant enzyme or plant structure that secretes the superoxide or produces the superoxide is this diagram. It's a molecule called NADPH oxidase, and these are in the plant cell membranes. And when the microbe is there, the plant can detect that and then hits them with superoxide. Okay, and so this is the structure that produces that superoxide, and it's in the plant membrane, plant cell root cell membrane. This is what these root cells look like when you see the protoplasts in them. And this is a root parenchyma cell, and you can see these arrows here. Those are those little spherical brown things. Those are the Those actually are the bacteria. Those are the protoplasts of bacteria. And they replicate very rapidly in chains. You can see there's a tiny one there. That's the youngest one. And if you go back, you see a larger one that doesn't stain as much. That's an older one. These larger ones break down. These younger ones, they stain blue because there's more protein in there. So these microbes are being degraded inside the plant, but they're also being replicated. They're also replicating. Fungi do the same thing. In fungi, these naked protoplasts are called mycosomes, the mycosomes. And this is an illustration from a paper by Atzat and Whiteside in 2014 that shows these kind of protoplasts that form when these microbes go in cells of plants. They're called mycosomes. We're not going to worry about those fancy terms, but they're protoplasts essentially all the same. Okay, really, these microbes are not going into the cell itself. They're not going in the cytoplasm. These microbes, once they get underneath the cell wall, so this is an illustration of that. The gray is the cell wall. Okay, so they go under the cell wall, and then they go into a space that's underneath the cell wall, but still outside the plasma membrane. So all this white, this outer line would be the plasma membrane. All this white would be the cell cytoplasm. So they're not actually in the cytoplasm. So the plant is secreting superoxide onto that microbe, but it's in this space just outside the membrane. That space is called the periplasmic space. Periplasmic meaning it's just outside the cytoplasm. Okay, so they're not really in the cell. They're just underneath the cell wall. Okay, this is a root hair, and it is stained for superoxide using NBT, a special stain, tetrazolium nitro blue tetrazolium, or NBT, which stains superoxide purple. And you can see these bacteria inside this root hair. And you see all these little dots in here. Those are all those protoplasts of bacteria. And the blue and the purple are the superoxide. So these plants are really hitting these microbes with superoxide. This is a different way of looking at these microbes. This is using autofluorescence. This is pseudomonads that will fluoresce when we look at them with the fluorescent microscope, and you can see these bacteria. It's the root hair. See the bacteria in those root hairs. They're autofluorescing. Now, that's their own fluorescence that we could visualize them. So we haven't stained them in any way in this case. It's all autofluorescence. You can see the tip of a root hair here, and you can see they're in that periplasmic space. It doesn't look like it necessarily when those are in the middle, but actually they're all of these protoplasts are around the periphery in that periplasmic space. That just shows another hair, shows another bacterium, and you see the microbes inside that hair. Well, the microbes are important for plants. All plants have them, and they control, we say modulate development of plants, but essentially they are controlling or influencing plant development. And they do that in a number of ways. One way is that roots will normally grow down with gravity. If we remove all the microbes away from the plant, the roots don't grow down. They lose that gravitropic response. Some roots will grow in the air. Some roots will just stay on the surface, but they tend not to go down into the soil. When the microbes are present, they will grow normally and grow down. Also, root hairs. If we remove all the microbes, no root hairs form on roots. The microbes are necessary in roots to have hairs developing on those roots. Okay, this is tomato. Normally, you would think that tomato fruits are sterile inside, but in fact, they're not. They carry microbes, and those microbes actually associate with the seeds in tomatoes. They will go around the seed, and some of them will actually go in the seed. And this typical tomato seed, all these little tissues have bacteria on them. And even if we surface sterilize and then we germinate this, there's bacteria that will come out of those seeds. So there's bacteria actually inside the seeds already. We've done lots of experiments with tomato. Okay, so this actually shows tomato. The seedling to the right, that's a tomato seedling, was treated with streptomycin. We surface sterilized everything off the surface using Clorox. and which sterilized everything on the surface. Then we treated with streptomycin for a period, antibiotic for a period to try to kill the bacteria. And in fact, you can see the seedling doesn't have bacteria, root hairs, no root hairs formed. To the left, there was no antibiotic treatment, and you can see hairs forming on those root hairs. This is typical. We've done experiments like this with lots of different plants. You see the same thing. Without those bacteria, plants don't develop properly. That's the point. And what happens, you have the case to the right here in which we remove, this is actually a root hair initial. So these roots will form these initials normally. But unless there are microbes that go into those root hair initials, they never elongate. So you just get a little bump on a root cell and that's it. But if there's microbes there, if there's bacteria in the seedling, then microbes will accumulate in the tips of those root hairs. And you see here, they will elongate. And you see all the way to the right, you see all those microbes in the tip of the hairs there. So without the bacteria inside the root cells, you get no hair development. Okay, why is the linkage of microbes to root hair elongation or to root cell growth? That's a major question. We've been working on that, trying to figure out exactly why. And one of my students who works on that question is this one, Ivy Chang. And she's done experiments with grasses and other plants. But with no microbes, you get roots like this. This is a grass root. And you can see there's no hairs on it. It's black because we stained for reactive oxygen. So you got hydrogen peroxide there. That makes it all brown. Okay, but with the microbes in which she put on, microbes back on, you can see all the hairs forming. And actually, if you look at the tips of those hairs, you'll see little spherical things. Those are actually microbes being injected out of those hairs. So microbes are ejected from the tips of those hairs. In fact, we think that's the function of the root hair, is really to move, and besides absorbing nutrients, to move those microbes out into the soil where they can acquire more nutrients. But one of our ideas, we're still trying to figure this out, is that microbes are secreting a plant hormone. And right now, the best candidate is ethylene. Ethylene, and ethylene is also microbes produce it using an enzyme called microbial ethylene synthase. Ethylene is also a plant hormone, and it is a growth hormone. When plants get ethylene, cells will grow. And so we think that the microbes are secreting ethylene and causing these hairs to elongate. And you won't be tested on this, so I'm not going to go into detail on this. This is the reaction we think is happening. The microbes are secreting ethylene. That's triggering the plants to produce superoxide and also triggering plant cells to grow. And the superoxide to ethylene combine, and it forms hydrogen peroxide. And that's why we're able to visualize this with the microbes in the cells with this hydrogen peroxide stain that we use called diaminobenzidine, because hydrogen peroxide is one of the products. So what we can also do, we have certain inhibitors that will inhibit this process. And one of them is this chemical called L-name, and it will inhibit ethylene formation. And when we inhibit ethylene formation, everything shuts off, the whole process. You get no root hair elongation, you get no superoxide formation, you get no removal of cell walls off of the microbes. Okay, this actually is stained for ethylene. It shows a root. This is a tomato root stained for ethylene. And you can see where the ethylene is. It's right here, this big brown kind of a orange looking area. Okay, that's where ethylene is produced. That's actually the area where microbes are entering the root cells, right in there, all over the surface of those roots. So that's where you get ethylene being produced. Also on the very tip, the tip of the root, that's actually the root cap cells. Those root cap is turning purple or blue. That's because ethylene is produced there. Microbes are entering those root cap cells also. And I'll show you that. Here, this shows a root cap cell. that sloughs off of the cap, and you can see the bacteria inside that cell. You can see them there, and this is stained for ethylene, so you can see the kind of bluish and purple color around those microbes. They are producing ethylene in those cells, hence the formation right around. You can see just a close-up of that. This is a root hair of the tomato, and it shows bacteria. We don't see any bacteria inside it, but you can see bacteria around it. And those bacteria, this was stained for ethylene. You can see they are producing blue. Again, that means they're producing ethylene using that ethylene stain that we use. Stain, by the way, is something called ammonium molybdate. They use the same stain to check for maturation in fruits, certain kinds of fruits like apples. This is a root hair primordium, and you can see the little blue cells. Those are the bacteria, again, inside there producing ethylene. This is a root hair of a grass, and you can see all the blue dots in there. Those are all the microbes producing ethylene inside that. This is a pseudomonad bacterium inside the root hair. Close up of that, all those microbes, blue ethylene produced. That just shows it again. Okay, so we're still testing that, and we've got a lot to do to confirm that's happening. But what happens if we remove the microbes? You get something like this. And this is Bermuda grass, no microbes present. We surface sterilize to remove the microbes, and you see there's no hairs. You see here, it's the older part of the root to the right, and there's no hairs at all. The no microbes, no hairs. We put the microbes back on, you see this. This is the tip of that Bermuda grass in the same experiment. We put Pseudomonas back on it, and you can see right away hairs form. Without the microbes, you get no hairs. With the microbes, you get hairs. They require these microbes for hair production. If you look at the cells, the root cells in that area where the microbes entered the cells, you can see here these little arrows, these brown, little brown. This is stain for this hydrogen peroxide using that DAB stain that we use. And you can see the rods. When they go in to the root cell, they have their rods, they have their shapes, they have their cell walls. It's only later that the reactive oxygen, the superoxide, will burn those cell walls off. But we've seen this many, many times in many, many plants. Okay, this shows actually a seedling root older, and you can see all the root hairs here, and you can see all the brown dots in there. Those are all the microbes. These hairs get filled with microbes, and the root cells, if you look at that, you see little brown dots in all the root cells. So all the surface cells of these roots get filled with microbes. That's a close-up of a root hair, and you can see these little brown spheres in there. Those are all the microbes. This is that pseudomonad in these root hairs of the Bermuda grass. They're filled with these microbes, and the plant will secrete these microbes out into the soil. And the way this works, I mean, you think of root hairs as just little, we all think of root hairs as just little static things that absorb nutrients, but that's not true. Root hairs are actually very active structures of plants. And this is a diagram of it. What you see in root hairs is there's this constant cyclosis or movement. The hairs vector or move around or circulation of the cytoplasm around and around and around the way I'm showing here in a counterclockwise or clockwise fashion, either way. But what happens is those microbes in that periplasmic space are actually moved to the tip, and that's where the wall is soft, and so they deposit, they get stuck out there in that tip, and they will push the wall out, so it's fat in some cases. So these root hair tips get really, really fat, but it's all filled with microbes because actually the hair itself is a little needle-shaped thing. It's skinny and tapers to a point. And then at some point, what happens is there's an expansion wave at the base of the hair, and it goes from the base all the way to the tip. And then it squeezes these microbes out, little pores. So it squeezes the protoplast out, little pores out to the tip. And then nutrients also go out, and then those microbes will reform their cell walls and their flagella and then swim out into the soil. So these hairs actively eject those microbes out. We think that it is potassium loading, or the plant will put potassium into the vacuole in these hairs, and then that will make water come in after the because of osmotic uh, pressure, and the water will then go in and it will cause this expansion and squirt those microbes out. That happens on a periodic basis. It's not continuous. Periodically, it will eject. We don't know what causes that. Here's one of the plants where we worked on. This is little sedge, again from Bonaire, on these rocks. It occurs on limestone rocks. Uh, it doesn't have much soil, but it does have lots of microbes. And this actually is a movie that shows the cyclosis. This is made from the reverse of a Petri dish. And so you can see these root hairs functioning, and this is a root hair. And you can see these little shadows going around. Okay, what's happening is this root hair is moving those microbes around the periphery. And we think that movement is important because what that does is that will actually break down any gradients that form between the root cell, the root hair cell, and the microbe, so that nutrients, if gradients form, it slows nutrients exchange. But with no gradients, nutrients flow fast between the microbe and the plant cell. We also think that movement, that cyclosis, is important because it breaks up those microbe protoplasts, so you get a bunch of smaller ones, and the plant essentially can clone those microbes by breaking them up like that. So it produces, where a few bacteria go in, many will go out and be ejected back out into the soil. The image below is stained so you can see the microbes. It's the same hair or similar hair from the same plant. And you can see now where the red are, the arrows I'm indicating, those actually are the microbe protoplasts inside the periplasmic space of those root cells. So this was done simultaneously so you could see the microbes. And then when I tell you that they're brown, those shadows are the microbes, you might believe that. But there they are stained, they just don't move. That's a different preparation. Okay, so the plant, in moving them like that in cyclosis, the plant breaks them up. And you can see here little clusters of microbes in a root hair. And this probably started from one microbe broken up. So you get many forming. Some of those are old and breaking down, but many form from the single one. So the plant clones and increases those microbes. This actually is hair. And you can see the cyclosis occurring. You can see the microbes moving to the tip. All these little white things kind of jostling around in that tip. Those are all the microbes. Those are the microbes there. They accumulate there. And in producing ethylene or whatever the hormone is, it causes that hair to elongate because in mass there, causes that hair to elongate, gets longer and longer. Those hairs will get long until all the microbes are gone. Then when they're gone, they stop growing. Till all the microbes are ejected, then they stop. And this you can see, so this is sped up a little bit, but not very much. This actually shows one of these hairs. You can see how fat it is. See the microbes there. You can see the microbes after they're ejected. They're forming their cell walls, but you can see inside, these hairs are actually filled with microbes. Not all of them are staining. These microbes will really fill these hairs. It's like this shows the ejection process. This is the student who was working on this. This is in tomato. And all the way to the left, you can see the swelling starting at the base of the hair. Here you can see in the middle, you see that swelling pushing it. You can see microbes coming out. And to the third, number 3 here, all the way to the right, you can almost see, I can see actually, looks like 3 pores where those microbes are squirting out of those hairs. And when they squirt out, initially they don't have cell walls, but they will reform their cell walls and then go back into the soil. So these are some images that show hairs. This left one all the way to the left shows whole seedling, and you can see this kind of clouded area around the root tip. That's where the microbes go in. That's where they're entering root cells. Then they stimulate the hair formation, and then the root hair kind of elevates those microbes out into the rhizosphere and then ejects them back into the soil where they can get more nutrients. So It apparently is a better process to move those microbes to the soil than it would be to just release them right on the surface of the plant where the nutrients have already been acquired. So the hairs may be important in this process for getting nutrients. The image to the right, all the way to the right, well, image to the middle shows bacteria coming out of those hairs, still photographs. And to the right, you see a couple of bacteria that are emerging from that tip. And you see little dots actually in that, little spherical things. Those are the protoplasts in those hairs. The nutrient flow in these hairs is actually away from the hairs. That is, the plants are squirting nutrients, exudates, out of the hairs following those microbes so that the microbes can rebuild. And you can tell that's happening because if you look at this as a yeast that's endophytic, a rototoral, a yeast, you can see, okay, they're coming out, there's little spherical things in there. Those are the protoplasts. But when they come out, you see they're tiny cells. And then as they age out here, you see they get bigger and bigger. So those microbes are growing. So there are nutrients that have come out with those microbes. That just shows that again. Okay, what happens to plants without the rhizophagy cycle? Okay, this is rice. Okay, you have, what we did is we removed all the microbes off rice, and then we put an endophytic bacterium back on. And see to the left here, you see where we put a pseudomonad bacterium on. You can see the cells get big. The seedlings get big. And to the right, you see rice without these bacteria. You see they're really not growing very well without the bacteria. And if you look at the soil, you see the plants here with bacteria, and you can see the soil is really adhering to that. There's a whole different relationship. Besides having root hairs, the soil will interact closely with those roots. Whereas if you remove the microbes, you see the little soil adherence. So the presence of the bacteria changes the whole relationship with the soil. It's very important. These microbes are very important for plant development. We have a lot of evidence that this rhizophagy cycle is really mainly a nutritional process. And that evidence comes from experiments that we've done. One of those is an isotope experiment, where what we did essentially is to take protein. We labeled that protein with isotopic nitrogen so that we could then measure it later, nitrogen 15. And then we put it into Petri dishes. And then we put seedlings with and without bacteria on that. And then we measured the amount of nitrogen that they're able to acquire. And this shows these Petri dishes that I talked about. And to the left, we have seedlings that have their bacteria. And you can see those roots go down and they're producing all the red. That's the reactive oxygen. So those are that oxidative interactions I was talking about. And to the right, we remove the bacteria and they go down into the medium to have protein in there with the 15N label, the isotopic nitrogen. But the seedlings are getting smaller. And then when we take those seedlings, we cut the shoots off and then we measure the amount of nitrogen that goes in, you get something like this. If you look to the right, this one, protein, no bacteria, you can see it goes up a certain level. And then if you look to the left, now this is with bacteria, and you see it's much taller. That's about a 30% increase. So we think that these microbes are worth about a 30% increase in nitrogen for plants. In other experiments done by one of our visiting scientists, he looked at wheat with and without the microbes. He removed some of the microbes from wheat, used those as test microbes, and then he sterilized the wheat and put the microbes on and compared it with microbes to wheat grown without microbes in terms of nutrient acquisition. Okay, so here to the left, this is without microbes. You can see the wheat is not very big, and you can see the roots are really stunted. With one of the microbes, you can see it has a huge roots. It's the same age and big shoots. These are exact same seedling in the same experiment with and without microbes. You can see it's making a big difference. And when you measure nutrients, measure nitrogen without bacteria, all the way to the left, you see no bacteria. You see it goes up to a certain point. Then with the bacteria, these are 3 different strains. You see it goes up about 30% more. So it looks like these microbes are good for 30% of the nutrients of nitrogen these plants are getting. Phosphorus, probably about 20% more for phosphorus. Potassium depends on the microbe, but you can see it's many, many more. Potassium, these microbes are carrying a lot of potassium to plants. Calcium, same about 25%, something like that. Sulfur, manganese, magnesium, micronutrients like zinc, iron. Okay, it's very important for all of these nutrients. You actually, I have one student actually who's working Ivy again, working on use of commercial microbes. And one thing she did is to check and see if these commercial microbes also can be used to benefit plants. And this experiment was involved celery. And basically, she put the microbes onto sterilized celery. And here you can see this is with no microbes there to the left. And she put the commercial microbes on, and you can see they stimulated hair development. And in fact, they went into the cells and into the root cells and stimulated development of those hairs. We've also done experiments looking at nutrient absorption, and they do also impact and increase nutrient absorption in many cases. This just shows one of these hairs, one of these root hairs with commercial microbe, a bacillus in it. You can see they're actually inside there. But it's important to test these products. So I mean, you can get these products, but you should actually test them yourself because not all these products are equal. Some are better than others. And the microbes in plants will also increase the quality of those plants and the quality of the nutrients that they contain. And this is one of my students that did actually work on carrots, and she looked at carotenoids with and without microbes, and she actually got microbes from celery, from another plant, other members of the carrot family. Put it that way, 3 different species, and then she sterilized the carrot and then put them on these different microbes on, and then measured carotene in those plants. So this just shows a carrot root hair. You can see the microbes in that hair. This shows roots. You can see the microbes in the carrot roots. And this shows analysis, carotenoid. This is alpha carotenes. And you can see the controls with no microbes. They go up a certain level. You can see bacterium 1 essentially was no better than the control. It didn't do anything. So it didn't help. Maybe it was not even endophytic. Bacterium 2 looks like it almost tripled the amount of carotenes that were produced. Bacterium 3 increased it slightly. So certain bacteria can actually benefit the nutrient quality of plants. Here's the same thing. You can see this is beta carotenes though, and you see that bacterium 2 again looks like it doubled or better than doubled, and bacterium 3 increased also. So in summary, these microbes, endophytes in general, and the microbes involved in the rhizophagy cycle help plants by absorption of nutrients from soils. And you can see here outcomes of the rhizophagy cycle, beneficial. One is nutrient absorption. Okay, another benefit is that plants have increased oxidative stress tolerance. They become hardier. And that's that oxidative interaction effect where plants, because they're oxidizing these bacteria, they have to oxidize their oxidation protection mechanisms, their antioxidants and so forth. And then they become hardier themselves. And finally, these microbes will also go away from plants and they'll colonize soil fungi, pathogenic fungi in the soil. They'll make those fungi leak nutrients, and they'll prevent those fungi from causing disease because they don't have the oomph or energy or nutrients enough to cause disease. And so these endophytes are really, really important in plants. So some take homes are that of this process and endophytes are: don't sterilize your seeds. Use seeds that have not been treated if possible. You don't want anything to affect the microbes on your seeds. Seeds will carry natural microbes, and you don't want to put something on there that hurts those beneficial microbes on the seeds. You want to manage your soils and the seeds to maintain your microbial communities. Soils, regenerative soils, organic soils are better than just soils that are sterile. You can use biostimulant microbes that you purchase. You just have to experiment with it. And endophytic microbes can improve the quality and the hardiness of plants. These are a couple of references that recent references, past couple of years, few years now, well, 21, few years now, regarding the rhizophagy cycle and endophytism, endophytes. Both of these are review articles. And these are all of the many people who contributed over the years to this work. And I'm happy to answer any questions that anybody has.
Thank you very much, Dr. White. Are there any questions?